On the previous sessions, we are talking about the hormones that is involved in the homeostasis of calcium and phosphate. So on previous class, we studied two hormones. So that is related with the calcium homeostasis. So that one is vitamin D, and the next one is that parathyroid hormone. So today we'll learn about the calcitonin, so another hormone, so that also involved in the regulations of calcium homeostasis. So like this parathyroid hormone, this calcitonin hormone, so it is also a, peptide, polypeptide hormone. So that means it is composed of amino acids. So it is a calcium regulating hormone, and it is also called as a thyrocalcitonin, since this hormone is synthesized by the parafollicular cells of thyroid gland. So so it is secreted by the this parathyroid, parathyroid cells of a thyroid gland. So it is also called as a thyroid C cell. So this thyroid C cell, it is derived from the neural crest. So generally this thyroid gland, it secretes two types of hormones. So one is that is a thyroid hormone, so which we studied in the earlier sessions, that is involved in a this basal metabolic rate of the body, and the which affects the entire metabolism of the body. And this calcitonin hormone, that is the another hormone, it is also synthesized by the thyroid gland, but it is involved in the calcium homeostasis. So while talking about chemistry, so it like these parathyroid hormones, it is also composed of a amino acid, so that is polypeptide in nature. So it contains 32 amino acids and having a molecular weight of uh, 3, uh, 3 3600 Dalton. So like you have seen this disulfide bridge on the vasopressin, oxytocin, so like that. Here also what happened, these 7 amino acids molecules, so they form a intrasulfide bond. So you can see here, so it's from a amino acid cycle, so that form a intrasulfide bond. And it contains a 32 amino acids. Among that, this thyroxine threonine, so that is the threonine amino acid, so it is most predominant in this polypeptide. So for the biological activity of this hormone, all inter-amino acids are necessary. So even a change in a single amino acids also, it loses a functional capacity. So when we studied during parathyroid hormone, so on that what happened, so the partial hydrolysis of parathyroid hormone also have a biological activity. So that means if the hormone is partially hydrolyzed and it contains this 30 34 amino acid sequence from the N terminal, then it is equally biological active as the full hormone. But in calcitonin, what happened? The loss of a single amino acid also, it loses the functional capacity. So for the biological active, it must contain all the 32 amino acids in the sequence. So synthesis, like other polypeptide hormones, it is also synthesized as a pre-pro hormone, and so it is synthesized by the gene transcribed. So generally it is so for the synthesis of this calcitonin, so two types of genes are there, that is a cal-Ca and cal-Cb. So that gene is transcribed and it's synthesized on the parafollicular cell of the thyroid gland. During a synthesis, these C cells, they generate a precursor protein of 141 amino acids, and after the trimming, that it when it's become a mature, so it contains a 32 amino acid sequence, calcitonin hormone. So this gene is also present in the other tissues, like in a the central nervous system, so which synthesize the protein containing the precursor, which can also synthesize a pre-protein which contains a 128 amino acids, and that produce another calcitonin type this related peptide, so which have 37 amino acids. in sequence. And this calcitonin gene related peptide, so it is involved in a this vasodilator. So regulation, so primarily this calcitonin is this the synthesis of this calcitonin is enhanced by the increased extracellular calcium concentrations. So it is just antagonist of the parathyroid hormone. So the synthesis of parathyroid hormone is stimulated by a low concentrations, low extracellular calcium. That is the low concentrations calcium in the serum. But for the this synthesis of the calcitonin is stimulated by a increased extracellular calcium concentration. So when there is the high concentrations of calcium in the serum, it stimulates the synthesis and release of calcitonin from the C cells of thyroid gland. So by this, you may know about the function of this calcitonin. So the function of calcitonin is just opposite of parathyroid hormone and the vitamin D. So this parathyroid hormone, it increases the concentrations of calcium in serum, and this calcitonin, it reduces the concentrations of calcium in the serum. And this parathyroid hormone, it also acts on the renal tubules and bone. So the actions on the bone and renal tubules is just opposite of the parathyroid hormone. So apart from these increased extracellular calcium concentrations, the release and the synthesis and release of calcitonin is also stimulated by gastrointestinal hormones like gastrin, secretin, and cholecystokinin. The significance of stimulating this calcitonin by these gastrointestinal hormones like gastrin, secretin, and cholecystokinin is that it prevents the hypercalcemia even we consume a high calcium diet. So actions, the actions of this calcitonin is similar with a parathyroid hormone. So like parathyroid hormone, it also shows its effects on the cell by stimulating the G-stimulatory protein coupled reactions. So that means, so it also acts on the hepatocellular G proteins from which this G stimulated protein, it activates the adenyl cyclase, and this adenyl cyclase in turn, it increases the concentrations of cyclic AMP in the cell. And by this cyclic AMP, so when this cyclic AMP is increased, so it activates the cyclic AMP dependent enzymes to elucidate its function. So the main functions of this calcitonin is it causes a rapid decline in calcium and phosphate in the serum. So generally for that, it increases the absorptions of calcium and phosphate from the serum to the bone, and it decreases the osteoclastic activity of the bone, so that this bone resorption is totally prevent, and then the releasing of calcium and phosphate from the bone to the serum is inhibited. Apart from that, it also inhibit the reabsorptions of calcium and phosphate from the renal tubules, so that what happen, it increases the these excretions of calcium and phosphate from the body, so that the concentrations of calcium and phosphate in the serum, so at higher than physiological doses, it decreases the number and activity of osteoclasts in the bone and calcium and phosphate tubular reabsorptions in kidney. So this calcitonin hormone, so it is a very important hormone in case of a bird and fish. However, in case of the human, the physiological role of this calcitonin is still unclear. So the physiological role in human is still unclear. So because, so if we remove a thyroid gland, so that which is responsible for the synthesis of a calcitonin hormone also. So even the removal of thyroid gland also, it does not uh, affects, or it does not create the clinical disorder relating to the calcium. So that's why this most of the scientists, they believe that there is a no role of the calcitonin in the calcium homeostasis, and it is this calcitonin is the only the it is the this immunological remains. So as we earlier mentioned that this one it's have a good relationship with the gastrointestinal hormones, so uh, which prevent the hypercalcemia produced by a food rich in calcium. So, however, it has a clinical point of view, it is of interest as a thyroid tumor marker. So if somebody have a this thyroid carcinoma, so then the level of calcitonin level is increased. And apart from that, in a higher concentration, it also acts as the inhibitor of the osteoclastic activity, so which prevents the bone resorptions. So because of that, therapeutically, it is also used for the patients having a osteoporosis. So therapeutic use, so that is, it is generally used, it is sometimes it is used for the treatment of some pain related syndromes. Apart from that, due to the its nature having a inhibitor of a bone resorptions, it is also used in osteoporosis. But the use of calcitonin in osteoporosis should be for a short period. If it is used for a long period, it may cause a different type of cancer. So it is related with the different types of cancer. So that's why the use of this calcitonin in osteoporotic patients is limited and short. It cannot be used for a long period. So this is that's all about this one, this calcitonin hormone. Now next topic, that is the regulations of intracellular calcium concentrations. So you all are familiar with a calcium homeostasis, that is the extracellular calcium. So that is the maintaining of this serum level of calcium, so which you have studied in during a first year. And generally this extracellular calcium homeostasis, that is maintained by these three hormones. So parathyroid hormone, this calcitonin hormone, and vitamin D. So like this extracellular homeostasis is necessary for the proper actions of skeletal muscles, that is the muscle contractions and proper actions of the heart and the other things like that. This like this extracellular calcium concentrations, this intracellular calcium concentration, so it is also tightly controlled or it is regulated. And however, the concentrations of the calcium in the intracellular is about 100,000 times lower than the extracellular concentration. So it is around 100 nanomole per liter. And the main functions of the intracellular calcium is as it is involved in the acts as the second messenger, and which is involved in the different activities of the cell, as it is involved in the regulations of the cell division, muscle contractility, cells motility, membrane trafficking, and secretions. So any change in the activity of a transporter so can change the cytosolic calcium ion concentrations rapidly and markedly. So generally, this the intracellular calcium concentration, so it is maintained by these calcium transporters, as well as some of the calciums they are also stored in a so organelles of the cells, suborganelles of the cells. So there are several processes that involve in the control of these intracellular calcium ion concentrations. So on that, so there's ATP dependent calcium pumps, so which supply the calciums from these extracellular fluids to the intracellular, and like that is sodium calcium exchanger. And also this intracellular calcium ion concentration is also regulated by this storage of calcium within the intracellular sites like this endoplasmic reticulum and mitochondria. So these calciums can enter cell through several types of these calcium channels, some of which are voltage operated or it is receptor operated. So to provide for a rapid influx in response to depolarizations or receptor stimulations. So the cell also maintain a large stores of calcium in microsomal and mitochondrial pools and in some cells in the Golgi. And this the regulations of that releasing of these calcium stores which is in a microsomals and mitochondrial pools. So that is done by the another second messenger, that is the IP3 inositol triphosphate. So I hope that all of you know about this. So these those hormones which work through the calcium. So most of them they activates the second messenger called as a diacylglycerol and inositol triphosphate. And this inositol triphosphate in turn it releases the calciums from the microsomal stores in the cell, and the increased calcium concentrations in the cell, they generally they individually or bound with the calmodulin, they shows the effects or physiological responses on the cell. So reuptake mechanism are also present so that intracellular calcium transit can be rapidly terminated by returning calcium to storage pools or pumping it across the plasma membrane. So the better example for this, you can see the this muscle contractions. So that is the binding of this sliding theory. So that means this binding of actin and myosin. So for that, you can you have studied these muscle contraction mechanisms. On that, what happened? So the increased calcium concentrations, so that which is released from the these microsomal stores, that calciums it bind with the troponin C, and so that this troponin and myosin they can bind and they can source the contractions. And for the relaxations, what happen, the calciums which is bind with the this troponin C is released, and again it back to the microsomal tubules. So this the increasing of concentrations and decreasing the concentrations of calcium ion within the cell is done by the releasing and reuptaking mechanisms. Okay. Apart from that, this releasing of this calciums from the supply of calciums to the intracellular by the extracellular fluid, and this transport of calciums from intracellular to the extracellular, so which determines the which maintain the concentrations of calciums within the cell. This is about the regulations of intracellular calcium. So now on this summary, that integrated role of parrot hormone, vitamin D, and calcitonin, so which you have studied in a first year. So, so generally to the maintain of calcium in a serum, so three hormones they regulate. So this parathyroid hormone, this calcitonin hormone, and this calcitriol. So that is the active form of vitamin D. And for that they generally involve on the three organs. So that kidney, bone, and intestine. So generally this calcitriol, this vitamin D, it majorly it acts on the intestine mostly. So when there is a high concentrations of plasma calciums, so high concentrations of plasma calciums, so it stimulates the calcitonin from a thyroid. See, there's a thyroid gland, and this calcitonin, so it inhibits the resorptions and enhance the mineralization, so that the calciums which is present in the serum, so that get deposited to the bone. And apart from that, this calcitonin it also increases the excretions of the calcium from the kidney, so the calcium concentration in the plasma get maintained. So if the calcium concentrations is low in the serum, then it stimulates the release of this parathyroid hormone from the parathyroid gland. And this increased parathyroid hormone, it causes the bone resorptions, so that the calciums and phosphate, which is stored in the bone that release into the serum. So it normalize the concentrations of the calciums in the serum. So apart from that, parathyroid hormone, it increases the reabsorptions of calcium from the kidney. So which decreases the excretions of calciums. And apart from that, this parathyroid hormone, it stimulates the formations of this calcitriol, that is the active form of vitamin D. And this in turn, this vitamin D, it increases the absorptions of dietary calciums from the intestine. And apart from that, this calcitriol on high concentrations, it help the resorptions of bone along with the parathyroid hormone. And it also increase the reabsorptions of calcium from the renal tubules. So the activity of this vitamin D. So the activity of vitamin D have is a dual nature on the on this calcium homeostasis. So on low concentrations, that is on a physiological concentrations, this vitamin D, it causes a mineralizations of a bone. So that's why when the vitamin D is deficient, then what happen? The persons this in the growing children's, they have a clinical disorder like rickets, and in adults they have a clinical disorder like osteoporosis. So on physiological concentrations, vitamin D causes a mineralizations of a bone. That means depositions of calcium and phosphate to the bone. But on higher concentrations, it aid on the resorption of bone. So along with the parathyroid hormone, it causes the resorptions of bone. That means it increases the osteoclastic activity. So this is all about the this integrated role of parathyroid hormone, vitamin D, and calcitonin on this calcium homeostasis. So from now, so we completed these hormones which is involved in this calcium homeostasis. So if you have any queries regarding to these topics, so that is in the calcium homeostasis, hormones involving in a calcium homeostasis, you can ask the questions now also, or you can ask by sending your questions on Viber or on other sites. So on the next class, we'll start this one, uh, hormones that is involved in the glucose homeostasis. So that means this hormone secreted by the pancreas. So on that, we'll study this one, insulin, glucagon.